alaikum. Hope you all are fine. So today we are going to start our chapter 3 on amino acids, peptides and proteins from Lehninger Principles of Biochemistry edition 8 and Lippincott Principles of Biochemistry. So in this lecture we will briefly introduce amino acids and their role in the formation of peptides and proteins. We will also look into the evolutionary perspective and early history of the amino acids. We will also discuss the basic or shared common structure of the amino acid, and there are certain exceptions to this common structure. We will also discuss them. So overall, this lecture will be an introduction of our chapter on amino acids, peptides, and proteins. So let's start. Okay, so what are basically the amino acids? Amino acids are basically the fundamental organic molecules. These are actually the first organic molecules to be found on the earth. So they are one of the important fundamental organic molecules, and you all know organic molecules are those molecules which have carbon and hydrogen in combined form in them. Being organic molecules, they are very important component of the one of the most and large you can say the macromolecules, which are called as proteins. So they are one of the most important component of the proteins. In fact, you can say these are building blocks of proteins, because in case of the proteins, tiny amino acids actually join together to make the proteins. And how it occurs, we can draw here. For example, this is a tiny amino acid. This is one amino acid, and then we have a second amino acid, and then we have third amino acid, and so on. We have different amino acids like this. Now these amino acids have this capacity that they can link with each other, and how they can link with each other and how they actually make the bonds with each other, we will look it in this into detail in later lectures. So this is one amino acid and it is connected to another amino acid, and then the second amino acid is connected to the third amino acid, and so on. These amino acids form various linkages with each other. Now this linkage which is actually formed between these amino acid, this linkage is called as peptide linkage, or you can say it's a peptide bond. And peptide bond, what is the nature of this peptide bond? It's a covalent bond. And from the organic chemistry, you all know that covalent bond always involve sharing of electrons. But there are certain other features of this peptide bond or covalent bond apart from the sharing of electrons, that it also involve a dehydration reaction. And dehydration reaction basically means it involves removal of the water molecules. And how it involves the removal of the water molecule and how this peptide bond is formed, we will look into this into the detail of this in just a moment. So this is how actually one amino acid is linked to the second amino acid, and then third amino acid and fourth amino acid, and so on. These amino acids are linked with each other and make this large string of the amino acids, which we call as protein. Now this is one of the simplest structure of the proteins. Actually these structures are very complex, and each protein consists of hundreds and thousands of the amino acids. Now this protein, once its basic structure, this long string of the amino acid, once this is formed, this protein now can take variety of shapes, or you can say variety of conformations, to attain its final active state in which it is fully functional. So we will all, when we will start the protein part of this chapter, we will see how the protein will take the different structures, like it can mold itself into the primary structure, into secondary structure, and then to the tertiary structures. And sometimes it also takes the quaternary structure forms to become fully active or to become the fully functional type of the proteins, because in this state, actually in this linear state, the proteins is not fully active, because it can be exposed to the various types of the proteinases. Proteinases are the enzymes which are found in the cytoplasm, which can actually destroy these peptide bonds and can basically denature this protein and make it functionless. So protein usually doesn't exist in this straight or linear form. It has to take the different conformations and states to attain its final active state. So coming back to the amino acids again, we have, okay, there are certain features of this peptide bond as well. We are not discussing them in detail in the introductory part, but we will discuss these certain features of the peptide bonds in a separate lecture on the peptide bond. So we will be discussing how the electric dipole is created in the peptide bond. That is very important. And how the this peptide chain which is actually formed, this protein or this polymer which is actually formed, how it can undergo the rotation and bending to attain the form final conformation or state to become fully active. And also we will be discussing the various angles such as dihedral angles like phi and psi angles, which are very important in determining the structure of the protein, in determining its conformation state, and to define the type of the peptide bond. So we will be discussing these points also regarding the peptide bond, but in the coming lectures. Now, as it's a introductory part of the amino acids, so we should also be focusing on the early history of the amino acids. Okay, in the early histories, there were two French chemists, Louis Nicolas and Pierre Jean. Both basically first discovered an amino acid in 1806, and that amino acid was named as asparagine. Why it is named as asparagine? Because it was obtained, or it was isolated from a source vegetable plant called as asparagus. So asparagus is a very common vegetable, and from that particular plant they isolated this amino acid in 1806. And later on in 1820, another amino acid was obtained from the gelatin. And gelatin, you also know that this is a kind of the protein which is commonly obtained from the animal collagen. And which animal collagen is used, which animals collagen is actually used, it is of mostly the pigs and cows. And what is the use of this gelatin? Mostly it is used for making the capsules. It is also enormously used in the food industry for making varieties of the food and nutrients as well. So from that gelatin they obtained a very important amino acid, and you can say the simplest type of the amino acid, named as glycine. And why it is named as glycine? First of all, from its source, as it is obtained from the gelatin, and also the in the Greek language, the word glycos mean sweet. So, due to its nature, it has been named as glycine. And apart from these two amino acids, these are the early amino acids which were discovered. Up till now, more than 500 amino acids basically have been discovered. And out of these 500 amino acids, 22 amino acids are more commonly found in making the biological structures. And what are these biological structures which they make? I have already told you. They make the structures which we call as proteins. So out of these 500 amino acids, or more than 500 amino acids, 22 are very important because these make our biological structures, our biological components, that is called as proteins. And proteins, you all know that one of the fundamental type of the organic molecule found in the body, your muscles, your body organs, your intestines, your gut lining, everything is made up of the proteins. So to make up these proteins, these are 22 building blocks which are actually involved. And out of these 22 building blocks, which we have up till now studied, there is one type of the amino acid which is not very commonly found in the proteins, and we are going to just see it in just a moment. But I have to mention here that majority of the teachers usually teach that there are 20 amino acids which are commonly found in the proteins, but there are actually 22 amino acids according to the recent studies. And out of these 22, usually the 21 amino acids are very commonly found in the proteins, and one is that which is very uncommonly found in certain type of the bacterial proteins. We are going to discuss it in just a moment. So in order to categorize these 22 amino acids and to make you better understand them, we are going to basically categorize them into the three main components. That is, we have, if we talk about the evolutionary perspective, we have the non-essential amino acids, essential amino acids, and the unusual amino acids. So, what are basically the non-essential amino acids? So, let me first of all write all the non-essential amino acids, and then we are going to write the essential amino acids list, and then we will also write the unusual amino acids list. So, you will be able to understand that which particular amino acids out of these 22 are essential for your body, are non-essential category, or the unusual category. Actually, non-essential and essential both are very important for your body, but why they're called as non-essential and essential, I'm just going to explain this. So non-essential amino acids, why we call them as non-essential amino acids? Because they can be synthesized by your body. This is very important. So these are the non-essential, that's why, because they're synthesized in your body, and you don't have to take these amino acids from any external source, such as from the diet as well, or as a supplement, or in the form of any other kind of the nutrient. So they can be synthesized in bodies of the living organisms. Okay, and essential amino acids are those which actually cannot be synthesized by your body. And if they cannot be actually synthesized by your body, definitely you have to take these amino acids from some external source, such as from your diet, or in the form of any supplement. You basically have to fulfill the requirement of these amino acids, because they are very important in making the proteins of your body. They are required actually by your body, but your body cannot synthesize them. So you have to take these proteins from in the form of the external sources, such as diet, or in the form of the supplements as well. And there are also two very unusual form of the amino acids. We are going to discuss them, and we will also see their names as well, and we will also study in which particular type of the animals proteins or organisms proteins these are actually found. So let me write the list of the non-essential amino acids. So in the non-essential amino acids, we have first alanine, then we have another name from A, that is arginine, then we have asparagine, which is actually the first amino acid that was discovered from the asparagus. Then we have aspartate, and we will see the structure of these amino acids in detail. They are actually a category of the amino acids based upon their argines, and we are going to discuss them in just a moment when we will go into the structure of the amino acid. So aspartate, then there is a cysteine. This is also a non-essential amino acid. Then there is glutamate. Then there is glutamine. And there is a very little, you can say, the minor difference in the structure of the glutamine and glutamine, and similarly the aspartate and asparagine. We will see the structures also. Then there is proline, serine, tyrosine, glycine. So these are almost 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. and 11. Okay, 1 2 3 4 5 6 7 8 9 10 11. So total 11 amino acids which are actually non-essential amino acids, and they can be synthesized by your body. And now let's take a look into the list of the essential amino acids. Here we have histidine, one of the most amino acids found in the DNA structure. Then we have leucine. Then we have isoleucine. And again, there is a minute difference in the structure of leucine and isoleucine. Both are isomers. Then there is another amino acid very commonly found, lysine, methionine, threonine, phenylalanine. It is very important amino acid because it contains a ring structure. Apart from all other amino acids, we do not have this sort of the structure. Then we have tryptophan, and we have valine amino acid. So now there are total 1 2 3 4 5 6 7 8 9. Total 9 amino acids are there which are actually essential amino acids. And there are also two more. So up to now 11 plus 9, 20 amino acids basically have been completed. 11 plus 9, 20 amino acids completed, which are usually you study in your textbooks, that these amino acids commonly are the building blocks of the proteins found in your body. But there are also two uncommon amino acids which are, one is selenocysteine, and from the name you can easily take an idea that it is the modified form of the cysteine amino acid. So we have already discussed here, this is a cysteine amino acid, and selenocysteine is just the modified form of the cysteine. Actually in the cysteine the sulfur is present, and in the selenocysteine it is replaced by the sulfur is replaced by the selenium, and we will look into the detailed structure of these amino acids in the coming lectures. And we have another amino acid that is called as pyrrolysine. So again, this is a modified form of another amino acid called as lysine, and it contains a pyrrole ring, and we will also see structure. And now the most important thing is that these are the very two uncommon type of the amino acids, and we usually don't talk about their structures and their occurrence, but this is very important here. You have to understand that these amino acids are also very important because selenocysteine type of the amino acid is commonly found in the enzymes of your body, and it actually makes, you all know enzymes are the proteins, so they actually makes the enzyme structures or the protein structures, and they're very important amino acid in those enzymes which have antioxidation properties. So mostly those enzymes which have the antioxidative nature, they are mostly found in those enzymes. So these are very important because if the enzymes are involved in the antioxidation, mean removal of the antioxidants in your body, so that is a very important kind of the enzyme to be considered. And mostly we usually don't talk about these amino acids because they are usually incorporated at the point of the stop codons in the messenger RNA. So when there's a translation process that is taking place, you know that if this is the messenger RNA, and here these are actually the ribosomes which are actually reading this strand. When there is a particular stop codon is here, and normally you know that when the codons are reading, the certain amino acids have been incorporated here. Okay, so when this stop codon actually reached, this is the end of the protein synthesis. So at this point usually we incorporate selenocysteine by the transfer RNA. So we will also look into this part in the coming lectures when we will discuss the protein translation, that how these amino acids are incorporated at the point of the stop codons in the proteins, so by the transfer RNAs. So now we have the pyrrolysine. Pyrrolysine is the very unusual type of the amino acids, and pyrrolysine containing proteins are very rare. So why they are rare? Because these are mostly found in the proteins of the bacteria called as archaebacteria. And archaebacteria, you all know that these are very ancient form of the bacteria, and they have very simplest form of the proteins, and these proteins are contain a very important ingredient, that is amino acid in the form of the pyrrolysine. So very rare, still very important, because from the archaeal bacterial proteins we can get the idea of the evolution as well, and we can take the idea of the evolutionary perspective of the various proteins. And you all know that according to the science, the every complex thing starts from the simpler entity, and from our knowledge that we originate from the bacteria. So to study the archaebacterial proteins and then the eukaryotic bacterial proteins, these are all the tiny pieces of the evolution which we have to join in order to basically study the more complex proteins which are today exist in the human body. So all these proteins which are found in the human bodies and in the bodies of the other living organisms, they all have linked with the ancient proteins found in the ancient bacteria such as the archaebacteria. And in these archaebacterial proteins, pyrrolysine is very important amino acid. So that's why this is very important with the if we say in the evolutionary perspectives. So this was all about the basic introduction of the amino acid. We discussed the common structure, common basically the points like definitely amino acids are the fundamental organic molecules, and how they make up the proteins, what are peptide bonds, certain features of the peptide bond. We discuss about the early history of the amino acids, their discoveries, their evolutionary perspective, commonly found 22 amino acids which make the biological proteins, and we studied as non-essential, essential amino acids, and we also studied the two very unusual type of the amino acids. In the next part of this lecture, we will be discussing structure of amino acids. Thank you so much for watching this video, and if you have any question, you can ask me in the comment box and I'll be happy to answer.